All right, Bio 402. So today we're going to be talking about the modern cloning method. And this modern cloning method was first established, or at least the person who is given credit for establishing this modern method for cloning, is a gentleman named Ian Wilmott. He was out of Scotland, and specifically he was out of a operation called the Roslin Institute, and this happened in 1996. And what Ian Wilmott is credited for is that he was the first to clone a mammal from an adult somatic cell, meaning an adult body cell. And don't forget, this is very different now from the cloning techniques that we saw in the 1950s. In the 1950s, those cloning techniques relied on having embryonic cells, and if you recall, embryonic cells are pluripotent for a time. And that means, if you don't recall, something that's pluripotent means they haven't yet specialized into their particular type of cell. And we also have another name for pluripotent cells, and that's called a stem cell. And I'm sure you guys have heard the concept of stem cells in the area of research where people are looking at curing people who are paralyzed. So anyway, that whole concept of pluripotency is a very important one. And if you go back and think about the old method, the problem that you have is, imagine for a moment, let's see if I can add an image here. Hold on, I'm just going to pause for a moment. Okay, so imagine that you had a cow, and shoot, did this just erase what I had said before? Seems like it's starting all over again. Oh well, we'll keep going. And that cow was particularly good at producing a ton of milk. Now, of course, you can only know that once the cow is an adult. And the problem is, if it's an adult, you can't clone an adult. Remember that the way that you clone things was from embryonic cells. And when you're an adult, all your cells have specialized into their particular type of cell. So, while it'd be nice, it's not really possible, or it wasn't possible until 1996. And so if you were making a clone, you had no idea about what that clone was going to be like in terms of its characteristics. So while it was a cool thing to be able to make a clone, it wasn't terribly useful to be able to make a clone. You want to be able to make a clone of something that's good, that you know works, that has the type of characteristics you want, not just some random little animal. Then it would be basically the same as just breeding dogs, essentially. All right, so anyway, let's take a look at what Wilmott did that made his work so interesting. Now I'm really worried that that first thing didn't record. I'm going to stop for a moment, go back in, and check to see that it's all functional. Hold on. All right, it looks like it did work, thankfully. So let's take a look at Ian Wilmott. Now, Ian Wilmott worked with a lamb, or a sheep, I should say. They were called Finnish Dorset sheep. And in fact, the sheep that became famous because of Ian Wilmott's work was a sheep named Dolly. Now, there's a ton of controversy associated with this whole thing. I'm not going to go into the details of that, but it's certainly worth a conversation for another day. Suffice it to say that Ian Wilmott's science, though it works and it made sense, his data were not good. And in fact, the things he did to back things up weren't very good either. Anyway, let's take a look at see what Ian Wilmott and Dolly look like. There's Ian and his pal Dolly sitting there. Now again, Dolly is an adult in this picture, right? This is Dolly. But keep in mind that this actually is Dolly version 2. So this is Dolly 2.0. I forgot, I can't really... oh, no, there it is. Okay, Dolly 2.0. That there was an original Dolly, and we'll just call her Dolly 1.0. if the original Dolly were around at the same time as this Dolly, then Ian could have had Dolly 1.0 hanging out right next to him on that side. Fortunately, Dolly 1 was actually dead at that point. Isn't that kind of sad? It'd be cool to have both the two clones hanging out next to each other, but alas, we never had that opportunity. All right, so how did he go about doing this? Let's take a look at the sort of protocols that he used, and I'm going to go to a new page here. So, we start off with a Dolly cell, one of Dolly's cells. And in fact, what's funny about this is that these were udder cells. Okay? And udder cells, also known as mammary gland cells. An interesting choice, huh? It turns out it's not like he really was thoughtful about this. He just happened to have a whole bunch of these mammary gland cells in his freezer, in his lab. And they were from Dolly, Dolly 1, who was long ago sacrificed for her cells. So Dolly was in a freezer, essentially, in bits and pieces. So they used her cells. All right, so we take this udder cell, and keep in mind that every single one of the cells that exist in the body of an organism have the exact same DNA. So all this green DNA that I have in there is completely identical to the green DNA that would be in any one of Dolly's cells, except, of course, for her red blood cells, which you guys know at this point don't have DNA when they're mature. So what we would really want to do is we want to take, let's see, this out of there, right? Because that we really care about the DNA, the nucleus here, okay? And in fact, this nucleus is going to be a very important thing, so we're going to keep that. Keep it around. And then what we're going to do is we need to find, don't forget, the other day I talked about the fact that if you have a typical egg cell, okay, that egg cell contains what are called factors. And those factors are what are responsible for the differentiation of cells so that they properly specialize into different tissues. Now, the only way that you can get factors is if you had an egg cell. Okay? And obviously this had to be an egg cell also from a sheep, or it's just not going to work. So we needed to find an egg cell, and it would be also a Finnish Dorset sheep egg cell. But then we needed to take out its nucleus, or enucleate it. Enucleated. So this was an enucleated egg, and we actually are able to, it's a good idea to start using these terms, but we call that, when it's enucleated, 0N. Now that doesn't say on, that says 0N. N standing for the amount of DNA, or sets of DNA that we have in that cell. Okay? So, then what Ian Wilmott did was he took this egg here. Let me just copy it and paste it down here. And then he took this nucleus, which he had removed from her, from Dolly's mammary gland, or the set from the freezer, frankly, and he was able to tuck this nucleus just inside the membrane of this enucleated egg. Now, keep in mind that if I were to, since I labeled the egg 0N, I should also make sure that I put a correct the correct symbols associated with the nucleus. And that nucleus is 2N. It has two sets of information, because keep in mind that Dolly was born from a mom and a dad as well, who both contributed their DNA. So she's a 2N, just like you're 2Ns, okay? Every single one of the cells in your body, except for your sperm or your eggs, are 2N cells. So we take this 2N nucleus, we tuck it just inside the membrane. So if I were to sort of draw a membrane around this egg cell, okay, a nucleated egg cell, tuck it just inside there, which is a tricky thing, for sure. Tuck it in there. Eventually, you could actually make this thing work. It would fuse it by, do I have yellow? Oh, good, I do, okay. you zap it with electricity. And I know that sounds really strange, but when you zap it with electricity, what happens is it allows that egg to fully take in that nucleus. So it looks something like this. So let me go ahead and copy this guy again. Oh, geez, I suck. Hold on, let me try this again. All right, copy, paste it. Oh, son of a... Paste, there we go. And then I'm going to put in the nucleus. So after zapping it, I was able to make that egg take in the nucleus. And then this too becomes a 2N cell. Okay, so now it's a 2N cell, but it's a complete cell. And don't forget, very importantly, what we have in here is that there are factors inside of this cell. Now, the problem is, while there may be factors, we got a problem with this DNA, because initially this DNA is completely differentiated, meaning it's been specialized. So in fact, all I've done is create an udder cell, or a mammary gland cell. So great, I put those into a petri dish and start growing them, and all I have is a bunch of mammary gland cells. Or even if I implanted them into a surrogate mom, she would produce a whole bunch of mammary gland cells in her uterus. That is not useful, and that is not a sheep, because the udder does not a sheep make. So we need to do something about this. We can't just leave it with this differentiated DNA in here. And that's where Ian Wilmott's work really started to take off and do its business. Because what he did is he added a very important step, which is that he starved the cells. Now normally, when you have cells in what is called culture, you have to make sure that you keep them healthy and happy. So you feed them, and you give them the right medium to float around in so that they can grow and be happy and take in nutrients and such. Otherwise, after a little bit of time, they're going to croak. So it'd be stupid not to make sure you provide them with the stuff that they need to be able to thrive. Well, it just so happens that that's exactly the situation you need if you want to have undifferentiation take place. Let me pause for just a moment. Okay, so here's a piano keyboard. And what I'm going to do is I want to generate sort of a strange metaphor that makes it easier for you to understand how DNA actually works here. So, as you know, DNA is made up of many genes. And I want you to think of each one of these keys associated with this keyboard as being a particular gene. So, you know you have the full keyboard at your disposal, but if you're one particular type of cell in your body, perhaps you only use this gene and this gene, right? And if you're a different cell, maybe you use this gene and this gene. So it's a very important thing to understand here, because if I hit this gene, and then this gene inside of a cell, and I'm not supposed to have this gene on, that could be pretty catastrophic. You could be creating proteins that are not going to work very well at all. Okay? Or if some reason your cell didn't produce this gene but only produced that one over there on the left, again, now a shortage of proteins may be a problem for that particular cell. So all cells know exactly which notes they need to play in order for that particular cell to do pretty well. But the cool thing is that as you differentiate, in order to make sure that mistakes aren't made, that cell will actually essentially remove keys or make them impossible to press down so that you don't make those mistakes. And only those particular genes, in this case keys on the keyboard, will be available for that cell to use. And it's a permanent thing. Those X's aren't something that come and go. Once the cell is differentiated, those X's stay in place. Okay? That's fully differentiation. Well, what Ian Wilmott did over here by starving the cells is he somehow got those X's to disappear. So all the cells, or whatever cell it was, suddenly had all the DNA turned on and available so that one could imagine being able to plunk any one of these keys, any one of the genes. Okay? That's the amazing thing, is being able to turn on all of the stuff so that it's all 100% available for a cell to use. And that's exactly what Wilmott did by starving the cells. He undifferentiated do I have room? Oh, phew. Undifferentiated the DNA in the cells, in this strange combination cell that he actually made here. So, now I have a cell that has undifferentiated DNA. Sorry, that was a mistake. Oh, I erased too much. I don't know, how do I gently erase? Oh, there it goes. Okay, so I have DNA that's now not differentiated, but it's undifferentiated. In other words, we've just created a pluripotent set of DNA. And don't forget that I added it to an egg cell that contains those factors, the same factors that are responsible for leading to proper differentiation or specialization. So what do? We take this wonderful cell that we just created that has been starved and now it's fully undifferentiated, and we plant that into the womb of a surrogate sheep. Now obviously that's not Dolly 1, that's not Dolly 2, that's just a random mom, a mom sheep. And we knew that she was ready, she could be impregnated with this funky cell thing. So we go ahead and we planted that into her uterus, and after whatever the gestation period is for sheep, I have no idea what that is. Anyway, after a certain period of time, voila, a baby is born, a lamb. And there's baby Dolly herself. This is Dolly 2.0, and this is Dolly's surrogate mom, who obviously is not at all related to Dolly because she's something like a Scottish blackface, I think was the name of her type of sheep. The same species, but they have different versions. Just like all dogs can mate with one another, but obviously we know they look very different. A Great Dane does not look like a Chihuahua. Well, same thing here. So there's Dolly 2.0. She was born, and clearly if we had Dolly 1.0 around to be able to compare her to, she would have been a complete and wonderful clone of Dolly 1.0. Pretty cool, huh? So again, it didn't take much here. Well, that's a lie. It actually took quite a bit to figure this out. I've really oversimplified this. But this new method allows you to ultimately create a clone from an adult cell so that you can have a whole bunch of Dolly 2.0s if you wanted. Or you could have a whole bunch of Darren Changs. Woo! All right, bye, you guys.